which is not very convenient, but well. Okay. So, ready, guys? Were you the one that asked the question? Did you hear me?
Partly, but could you repeat it again, please?
Yes. So you asked, I was just trying to remember your question. So you asked if then there will be more soluble at alkaline conditions. And I said yes, but depends on the species. If, for example, soy protein in our lab is extracted at pH 7.5, and you can get good protein solubilization. Of course, with especially plant proteins and soy and legumes, with a higher pH, you have higher solubility because of the ionization of other amino acids like the OH containing and the sulfur containing, sulfide groups containing. So yeah, does that answer your question?
Yes, thank you.
Okay. Was there a second question or was that it?
Yes, I did have one trailing question. So I realized that in legumins and viscilins, we talk about subunits, acidic and basic, but we never said that for whey and caseins. We always said like pockets. So why do these proteins have subunits versus some other types don't have, or maybe how a subunit is defined?
Yeah, it's coming. It's coming in the next slide. I'm going to talk about subunits. So if you think of casein, it's actually also a quaternary structure with multiple proteins associated together. So basically, when we say subunit, it's a protein molecule, but it doesn't exist on its own. It associates based on its characteristics and surface properties. And we're going to talk about that. And that's why legumin is a polymer. It's not one protein. It's a polymer of different subunits of protein molecules that are associated together. And we'll talk about that. So it's coming. Yeah, so in animal, like whey protein, because of its surface hydrophilicity, how highly hydrophilic it is on the surface, so it doesn't really form quaternary structure unless the pH is around the isoelectric form. So they exist as monomers. But it all has to do with the surface properties of the protein. Okay, all right, so looking at the legumin again, and speaking of the subunits, so the legumin molecule, which is 11S, is composed of what we call 6 monomers. Okay, and each monomer, so if you look here where I'm putting my cursor, so you have the black sphere and the blue sphere. The black sphere and the blue sphere, each one of them is a protein molecule we call a subunit, but they're linked with each other by disulfide linkages. Each monomer has 2 disulfide linkages. So I have 4 cysteine residues, and they are associated by disulfide linkages. So this is the monomer here. It's a combination of, where is my cursor? It's a combination, it's a subunit that is acidic and a basic subunit. So the acidic is a larger sphere because it's simply a larger molecule, have higher molecular weight, ranges between 30 to 40 kilodalton, depending on, again, the species or the variety. And the smaller one is the basic, and it has an average molecular weight of 18 to 20 molecular weight, again, depending on species and variety. So each one of these spheres is a subunit. And when they are formed in a disulfide bondage, so they have 2 disulfide linkages, they form a monomer. So this here, what you're looking at is 3 monomers. So this is 1, and then 2, and then 3. So the red bar represents disulfide linkages. The black bar represents hydrophobic interactions. So you have here 3 monomers associated by hydrophobic interactions. So another characteristic is even within the monomers, you have homology, sequence homology between the monomers, because the monomers are made of these basic and acidic subunit, but even the acidic subunit or basic subunit, there are multiple genetic variants of it. So each subunit would be slightly different than another acidic subunit, and each basic subunit will be slightly different than another basic subunit. So there is some variation between the monomers, hence the differences in molecular weight. When we run the gel, for example, in juicing conditions, we see several bands representing the acidic and the basic subunits. Okay, so this is a trimer via hydrophobic interactions, and each trimer is a hexagon, so because you have 6 subunits, and then they form the hexagon, the trimers form the this particular shape where you have 2 trimers on top of each other. Well, it depends what's top, what's bottom, but I mean 2 trimers interact with each other via hydrogen bonding and electrostatic interactions. So these dotted lines represent non-covalent interactions, mostly hydrogen bonding and electrostatic interactions. And then there's a lot of random coil, high percentage of random coil in general compared to beta sheet and alpha helices. So if we run the gel, protein isolate on a gel, we see a lot of high molecular weight polymers. And once we run them under juicing conditions, we kind of broke down the disulfide linkages here. So each of these subunits is migrating separately now. And also in STS, you break hydrophobic interaction, hydrogen bonding, electrostatic interaction. So you break that too. So you see those individual now subunits. And these individual subunits represent, well, we didn't talk yet about beta-conglycine, which is the vessel, and this is soy protein. So these are the acidic subunits of legumin, in this case glycine because it's soy. And this is your basic subunit. It's around 20. This is around 35 to 40. And then there are you see multiple bands here because you have multiple variants of the acidic subunits. So this is an animation produced by Fan, one of our students. That is very curious individual. So over quarantine, she took a course and learned to animate protein structure, and she gave me those figures, which is really nice, where it shows your acidic and basic chain, and then the linkage, the disulfide linkage between the 2 subunits to form the monomer. And then you have your trimer. So this is one monomer. This one here is another one, and this is another monomer. So this is your trimer and another trimer that form a hexameric glycine or legumin 11S. And this is what I mentioned, that you have variants also of these individual subunits. And there are a few that have been classified based on proteomics type of work and sequencing into 2 different groups. And within each group, you have those different variants, A1A, A1B, A2. These are 3 different variants of the acidic subunits, and then different variants of the basic subunits. And then another set down here of another group of different variants. And it's showing you that the homology within these variants within one group is over 84%. So they're very close in amino acid composition and sequence, up to 84%. And these two groups are different and with homology less than 48%. With some additional differences that you really don't need to memorize, or you don't even need to memorize the different subunits, just that, I want you to know that there are different variants. So when you look at a gel, you have multiple bands. We call them all acidic and all basic because they're basically, you have multiple variants of these different subunits. So Vicin, it's a smaller polymer than legumin and has a fraction of 7s sedimentation coefficient and average molecular weight of 180. So it's not like exactly 180, this is an average. And again, it varies from the different variants and subunits within each polymer. And it's a glycosylated fraction. That means some of the serine and threonine and amino acids that can be glycosylated are glycosylated post-translational modification. Does not have a lot of sulfur-containing groups, so there is no disulfide linkages. And it is rich in amides of acidic amino acids, but are low in basic amino acids. So compared to legumin, which is high in arginine, for example, it is low in arginine. See here. So the Vicin is a trimer. So you have 3 subunits, and these subunits are associated by hydrophobic interaction mostly, so no covalent interactions. And the average molecular weight is 60, but that doesn't mean that each one of them is 60 kilodalton. The average of them together is 60 kilodalton. There's a lot of sequence homology as well. And this is the distribution of the secondary structure. So on the gel, when we run them under SDS and, well, reducing it doesn't matter because there's nothing to be reduced in terms of disulfide linkage. So you see those main variants or subunits, the alpha prime, alpha and beta subunit of beta-conglycinin, and they vary slightly in molecular weight, which we'll see them separated as 3 individual bands around these average molecular weights, 72, 68, 52. So the average is about 60 kilodalton there. So all 3 subunits, like I said, are acidic because they're high in acidic amino acids and low in basic amino acids. And the alpha prime and alpha are mostly on the exterior of the trimer, and the beta is more hydrophobic, is in the interior of the trimer. So kind of this is a representation of the trimer here. So you can see that they associate in a way where the beta subunit resides most of it in the interior of that structure. So this is animation from Fan. It's showing the trimer and these little groups here and here and there. These are the glycosylated groups of the each subunit, basically. And this is 7S in soy is beta-conglycinin, in peas, Vicin, and other pulses is Vicin. Known as Vicin, just Vicin. Again, there are different species of the trimers. Some trimers contain 1 alpha prime, 2 beta in one trimer. Others have all 3 different subunits in one trimer. So they vary. So they've been identified as 7 different species of these trimers. The convicillin is present in pea and also chickpea, for instance. So in pea and chickpea you have legumin vicillin and convicillin, and this is also a trimer, and it can be up to 290 kilodalton. Again, it's a range, it's an average. And yeah, so in P, you'll find vicin present as well as vicin. This is an interesting image that made me think of Holly and her fractionation. So Holly, you might want to look up these articles. They separated convicillin from vicillin fractions, even not just vicillin from legumin, which is interesting. But anyway, so this would be convicillin on the gel. These are all different subunits of vicillin. Okay, so there are different molecular properties that I will be talking about to understand how they associate and dissociate the polymeric forms of legumin and vicillin, how environmental conditions impact the association and dissociation, some solubility differences between these fractions as well as between the subunits, impact of pH, ionic strength, heat stability, and susceptibility to enzymatic hydrolysis. So we will talk about all of these differences. In general, there might not be, I don't remember a slide specifically on enzymatic hydrolysis. In general, globular proteins resist hydrolysis. So actually there will be a slide on enzymatic hydrolysis of soy protein in general. I just remembered. So yes, so we will cover all of these. We'll start with the association dissociation. So again, as I mentioned, for example, the legumin as well as vicillin. So the legumin has multiple interactions within that one polymer. So you have the hydrophobic interaction, the disulfide linkages, the ionic interaction, the hydrogen bonding. So these are all impacted by the environmental conditions. You have how are they impacted by salt concentration, by pH, because they both change the ionic interaction with water and with each other. And pH changes the charge, and temperature causes the maturation, and protein concentration would also enhance formation of larger polymers because of the shorter distances between the molecules at higher concentration. Presence of denaturing solvents and reagents such as urea, SDS, sodium hydroxide sulfide, and presence of reducing reagents or conditions will all impact whether we have a large polymer, a 15S at higher concentration at specific temperature and ionic strength and pH, you get more association. So you get this larger polymer. And then under other conditions, the 11S can dissociate, and the 7S can also dissociate as well under certain conditions to smaller sedimentation coefficient fraction. So here's an animation of that. So this is your hexameric legumin. If we change the ionic strength and pH in a way that changes the ionization, it might interrupt with interrupt the hydrogen bonding and the ionic interactions or the ionic bonding. So you might get a trimer. So this will break into that trimer. With additional pH, like a very high pH or very low pH, you get a lot of repulsion. So then you end up having a monomer. Or if you have a denaturance, like if you have SDS, for example, this is going to break the hydrophobic interactions, and then you end up with your monomer. With high temperature, without the reducing agent, the subunits, even though they're linked, they will open up and denature. So you will have unfolding. If you add a reducing agent, these monomers will break down into basic subunit and acidic subunit. And then with heat, also, or extreme denaturation conditions, these will be denatured. The tertiary structure will be denatured. So you have, when we talk about association, dissociation, we're really talking about the quaternary structure changes in quaternary structure. When we say denaturation, you're having a change in the tertiary structure of these individual subunits. Okay, so here are examples of pH and ionic strength under which conditions you will have a hexamer, a trimer, a monomer, etc. You don't need to remember those. It's just illustrating that under different ionic strengths, the mu is a representation of ionic strength. So at different pH and ionic strengths, you have different association and dissociation. As well as the dissociating solvents are mostly urea and STS. And at high concentration of acid, you get more denaturation and dissociation. And also this is the denaturation temperature of the individual subunits. So glycinin or legumin is close on average to 90 degrees, and beta-conglycinin or vicin, they range between 75 to 82 degrees, around that range. Again, it's a range. This is kind of an average, but it's a range depending on the species, depending on the variety. What we're looking at here is a differential scanning calorimeter. The different types of DSCs softwares produce either a positive peak or a negative peak. Some DSCs, like the one that currently is working, this is an older DSC that we had that is down at the moment, gives you a positive endothermic peak. But basically it gives you the same information, the denaturation temperature and the enthalpy. That means the energy it took to denature that protein in this program where you put the sample in and then you ramp up the heat until you get denaturation. So this is a soy protein isolate we prepare in the lab. So we see two distinctive peaks, one for legumin over here and one for vicin. That means glycinin. This is an isolated fraction of glycinin, and this is an isolated fraction of beta-conglycinin. So the glycinin denatures at a higher temperature because of the fact that you have a more compact disulfide link molecule. So it requires more energy to denature. So the vicin or the convicillin or beta-conglycinin denature at lower temperature. What I'm showing you here is a commercial SPI. When you don't see, this is a reminder, when you don't see any endothermic peak, that's an indication of its completely denatured protein. All right, this is our current DSC. It gives you a negative peak. And the same thing, we calculate change in enthalpy, and also it gives you the denaturation temperature. So here you can see onset and end-set, but then the peak is your denaturation temperature, and then you have the enthalpy here in joule per gram of protein, you correct for protein. So this is P, for example. In P, you have more of vicin, convicillin, and less of the legumin. So we don't see a very nice separation in the peaks, and the legumin component is a smaller peak. So when we integrate this kind of like under the curve, this area, and we integrate this, you have more vicin, convicillin. They don't get separated because they're close in their denaturation temperatures. So we don't see them as two separate peaks. So in peak, you have more of the vicin and less of the legumin. So you see this shape. In soy, you have more of the legumin and less of the vicin. So you see a smaller peak for vicin. This is, for example, a chickpea. I get this from Brigida. She had ran those samples. So in chickpea, you have more legumin than vicin in chickpea. So you see more pronounced peak for legumin around, similar to soy, around 90, well, a little higher than the denaturation in soy, which is 92 to 95, here around 100. And then the Vicin is around 82 degrees. But again, the difference is legume denatures at higher temperature than Vicin. This is a little differentiation too between chickpea, pea, and soy. In here we have pea, and this is soy. So in pea, you have quite a bit of the acidic and basic subunits of the legumin, which is the glycinin. If we look at the legumin, and the acidic, the alpha is for acidic and the beta is for basic. You see that you probably have, not probably, you have lower legumin proportion compared to convicillin. The Vicin here, a lot of bands here for Vicin and more bands here for Vicin as well. So that is a differentiation in different subunits among legumin and Vicin from two species. And here's the third species. This is the chickpea. Also, this is run by the Gita here that shows you also differences. This is acidic, basic subunits, and that are more, you have more legumin than you have in the pea. Okay, I'll go for 5 more minutes and then we'll stop. In terms of solubility, it follows the bell shape that we talked about in terms of plant protein compared to whey protein. Around the isoelectric point, we lose a lot of charge and you have high hydrophobicity on the surface. Therefore, you have more protein-protein interaction and less solubility. Whereas at acidic pH, you have a little bit more charge and you get higher solubility. At basic pH, you get even more solubility due to ionization of other groups. So, oh, that's a recap question. This means that I probably should stop here. Okay, I'll let you go 4 minutes early. It's fine. All right, I will record the rest of this and hopefully start the serial proteins lecture. Make note that it would be about equivalent to 250 minutes lectures. You can pause and rest. You don't have to listen, but I'm not going to pause. I'll lecture for 50 minutes at the same time. Okay, I'll see you. I won't see you till April 21st in 3 weeks. All right, take care.